say, okay, well, what would happen between having a genetically programmed immune deficiency and having IBD, Crohn's disease? So to me, this is now the start of the hard work. You have to ask, what are the kind of bacteria that could exploit this pathway when it is defective to engender, in the long run, a chronic inflammatory disease? So, one bacteria, well, among bacteria, you'd have to ask yourself, well, what are the kind of classes of bacteria? So a very simplified view of bacteria could be to say that bacteria can be either on the outside of cells or on the inside of cells. We call them extracellular or intracellular. So extracellular bacteria are things like Staphylococcus. There's probably Staphylococcus on some of your hands. Many people carry it in their noses, and they just sit on the surface of things. And they cause all kinds of different diseases. You're aware of skin infections and stuff like that. There was a very famous outbreak of Staphylococcus aureus among football players that was published in the New England Journal of Medicine because they were able to show that it was crossing from one team to the other team, especially the linemen who seem to have the most physical contact. Those are extracellular bacteria, and to date, the evidence is that the Crohn's genes are not particularly important for extracellular bacteria. And then there are intracellular bacteria, things that live inside of our cells. Now, we study Mycobacteria, but they are by no means the only intracellular bacteria. Listeria, of which you're probably aware from the Maple Leaf Foods outbreak, is an intracellular pathogen. And Mycobacteria are also intracellular bacteria. Mycobacteria are named for the cell wall, so they're made of mycolic acids. That's why they're called Mycobacteria. Mycobacteria include two organisms that have already proven themselves to cause chronic inflammatory diseases, tuberculosis and leprosy, and they are therefore a subset of the intracellular pathogens. And it's very important when you've got your candidate to remember that your candidate's not the only thing out there, and you have to ask about other things that are comparable and how your candidate or your hypothesis fits against alternative hypotheses that may also fit the data. So, we've asked two questions about Mycobacteria and NOD2 because we're a Mycobacteria lab. The two questions quite simply are, is NOD2 important when you have a mycobacterial infection, and is NOD2 specific when you have a mycobacterial infection? So there's going to be a little bit of science here just to let you know of what sort of, the basis of what I'm talking about. I don't want to bore you at this point, but I just want to go over a couple of these features. So the first paper, we took mice that don't have NOD2. So they're not humans, I accept that, okay? And we did not look for Crohn's disease. We're not even looking at an intestinal infection. We just asked, when mice don't have NOD2, how do they handle a mycobacterial infection? We put tuberculosis in their lungs. We put the BCG vaccine that some of you may have received in your childhood. We put that into their lungs. And we monitored them for shorter and longer periods of time, and we found that they have defective innate responses early to the mycobacterial infection, and that they made defective longer-term adaptive immune responses, that their lymphocytes did not recognize the mycobacterium when we took them out of the mouse and tested them in the lab. It was as if they were somewhat unaware that they were being a mycobacterial infection. So, accepting that we used TB and we studied the lungs, you are going to say, this doesn't tell me much about Crohn's disease, so I'll say it first. So, what else did we do? We asked a different question, which is, when NOD2 is disruptive, is the thing that we observed with TB a common feature of any bacteria, or is it something that is kind of unique to certain bacteria, right? You could say, well, if NOD2 is involved in bacterial recognition, then when NOD2 is missing, you're not going to recognize any bacteria, and that's just going to be a very generic defect that you just can't deal with them. Or is it certain bacteria that you have a specific problem handling? So what we did is we got a dozen different bacteria, and we asked us, how does the absence of NOD2 matter for all these different bacteria? And the reasoning was, if we can figure out where NOD2 matters the most, then we would infer that defects in NOD2 would be most important, okay? You see the reasoning there? So what we did is we asked ourselves, when we have mice that have NOD2 and mice that do not have NOD2, and we take their cells and we expose their cells to Mycobacteria, Nocardia, Rhodococcus, Listeria, Staphylococcus, E. coli, Salmonella, and we ask, how important was NOD2 for all these different bacterial exposures? We come up with a family tree of the bacteria. So this is based on the genetic similarity of all those different bacteria. And it turned out in these experiments, consistently and reproducibly, NOD2 was important for these bacteria up here. And NOD2 was of no relevance when you threw these different bacteria there. Okay? So that's what we call NOD2 dependent. When NOD2 is missing, you have impaired responses. And down here, we call it NOD2 independent. When NOD2 is missing, nothing happened. NOD2 was irrelevant for the recognition of these bacteria. Okay, so why would that work, and how would that work is what we asked. So here's the science. So for those of you who've taken undergraduate microbiology or biochemistry, the bacterial cell is made up of a wall called peptidoglycan. This ring here is called N-acetylglucosamine. You can buy glucosamine at your pharmacy. And this is called N-acetyluramic acid. And these guys, these rings go together, and then there's a little chain here of amino acids, and then there's another glucosamine and uramic acid here that sends its chain, and all of these link together like a spider's web that surrounds the bacterial surface. Turns out that NOD2 recognizes this. It recognizes that ring with these two amino acids. And that thing, you don't have to know much more about it, that thing is called muramyl dipeptide, MDP. And different groups consistently around the world all say that NOD2, when it works, recognizes MDP. And in Crohn's disease, people have defective recognition of MDP. It's not exuberant, it's not hyper-response, it's defective. If you have the NOD2 mutation that predisposes to Crohn's disease, you do not recognize MDP. So the question we asked is, do all bacteria make the same form of MDP? Because if every bacteria makes MDP, NOD2 should matter for all bacterial exposures. And the answer is, if you look over here, here are the NOD2 dependent bacteria, and here are the NOD2 independent bacteria. It turns out that the bacteria that are NOD2 dependent have a specific enzyme and they make a different form of MDP. This is called N-glycolyl MDP, and all of these bacteria do not have that enzyme, and they do not make that one, they make N-acetyl MDP. So in chemistry, this is extremely subtle. We're asking only about converting a group here by adding a single oxygen and hydrogen, OH group. And then there, you, instead of having N-acetyl, like vinegar, you have N-glycolyl. And that little difference converted the bacteria for which NOD2 didn't matter to the bacteria where NOD2 did matter. We then decided to just knock out that enzyme from the bacteria, and that's exactly what we saw. This is the inflammation that you make in cells, and when they are not infected with bacteria, they do not make TNF. You'll remember that the treatment of Crohn's involves anti-TNF agents, which is a marker of inflammation. So cells that are uninfected make no TNF. The mycobacteria that has the wild-type enzyme makes this much TNF, and when you knock out that enzyme, you make less. When NOD2 is missing, you make less. Here, we took the mycobacteria where we got rid of that enzyme, so it only makes the acetyl form, and then NOD2 suddenly didn't really matter too much. And when we put the enzyme back into the bacteria, NOD2 once again became relevant. So NOD2 doesn't explain all of the recognition of the bacteria, but this is what we call NOD2 independent. In these bacteria, it doesn't really matter if NOD2 is there or not, because you get the same amount of TNF. While in this bacteria and in this bacteria, NOD2 matters. When NOD2 is there, you make more TNF, and when NOD2 is missing, you make less. So we showed that you only observe NOD2's importance when the bacteria has this bacterial enzyme. Okay? NOD2-dependent recognition happens when MH is present. So NOD2 is not a general sensor of all bacteria. Rather, NOD2 is a specific sensor for a certain family of bacteria.